Yo, tell me when to. Okay. Yo, 1, 2. Me. Yo, tell me when to. Yo, 1, 2. Me. Yo, tell me when to. Yo, 1, 2. Me. Yo, tell me when to. Yo, 1, 2. Me. Yo, tell me when to. Yo, 1, 2.
When to study transcription? Right now. Now's the time you should study. So let's get started. Now, I'm sure you guys remember. Transcription produces an RNA that's complementary to one of the strands of the DNA. And one of the two strands of the DNA serves as the template to make the RNA. Now, I'm sure you remember that transcription is different from DNA replication. And it's different for two reasons. One, the RNA does not remain hydrogen bonded to the DNA template. Right? If you remember with DNA replication, the new strands that are made, they remain hydrogen bonded to the nitrogenous bases of the two strands of the DNA that get split apart, right? And that's why it's a semi-conservative model. One strand is the parent strand, and a new strand remains hydrogen bonded to it, and the other strand is a parent strand, and the new strand is hydrogen bonded to it. But again, in transcription, the RNA, it does not remain hydrogen bonded to the DNA template. Okay, number two, the other reason why transcription is different from DNA replication. The RNA that you generate during transcription, it only comes from limited regions of the DNA. We're not transcribing the entire length of the DNA strand. That would be millions and millions of base pairs. It's not like that at all. We're just looking at short pieces of the DNA where the genes exist, and we're transcribing those pieces, okay? Now, so again, remember the DNA is used as a template to make the RNA transcript. The RNA is single-stranded, right? So it folds up on itself. Remember C to G, A to U, okay? It takes on weird, complex shapes. It doesn't say strung out like a spaghetti noodle or something, right? Okay, now, the RNA polymerase. How does this enzyme work? Well, just like DNA polymerase, it catalyzes in a 5' to 3' direction, right? And it uses ribonucleotides, not deoxyribonucleotides, not those little DNTPs, right? It just uses ribonucleotides, and it uses ATP, CTP, UTP, and GTP, okay? But the RNA polymerase, it is quite a bit different from DNA polymerase, okay? Why? One, like I just said, RNA polymerase catalyzes the addition of ribonucleotides, not the little DNA deoxyribonucleotides. Two, RNA polymerase does not require a primer. I'm sure you guys remember we talked about DNA primase and how it had to lay down the RNA primer before the DNA polymerase could come and add new nucleotides to the 3' hydroxyl. Well, RNA polymerase, it doesn't need to do that, okay? And number three, RNA polymerase can only make a mistake. DNA polymerase, as you know, can make a mistake in about 1 in a million. And that's because it has such a great proof reading mechanism in the 3' to 5' direction. RNA polymerase, it does make mistakes, and it makes a lot more mistakes. Generally about 1 in every 10,000 nucleotides that it copies, 1 in 10,000. Okay? But if you remember, we talked about this. Okay, why is this an issue? Well, during DNA replication, if a mistake is made, that could get passed on for generations and generations and generations and generations. Okay, snap out of it. But with RNA, and made in transcription, you make a mistake, so what, right? You're going to be making tons of these transcripts. Yeah, maybe you're going to make the wrong protein. Who cares, right? That protein will be defective, but it's not going to get passed on to the offspring. So it's not that big of a deal, hopefully. Okay? But, so even though RNA polymerase, uses ribonucleotides, it doesn't need a primer, and it makes mistakes, it still can proofread a little bit. And the proofreading is just like with DNA polymerase in the 3' to 5' direction. Okay, so what are we making with this RNA polymerase? Right? There are several types of RNA that we can make. We could make messenger RNA or mRNA, right? That's the one that we think about the most. We could make small nuclear RNA, that's little SN RNA, which directs the splicing of the pre-mRNAs that are made. Okay, they kind of work together. We could make ribosomal RNA, little r RNA. It forms the core of the ribosomes. We could make SNO RNA, which also plays a role in the splicing mechanism, the small nuclear RNA. Or we could make transfer RNA, tRNA. They serve as those little molecular adapters between the ribosomes and the amino acids that are just floating around out there in the cytoplasm. Okay? So there are lots of different types of transcripts that we can make. Okay, let's talk about, remember we talked about how we don't transcribe the whole genome. Instead, we just transcribe little pieces of it, right? What are those pieces called? Well, they're transcription units, okay? So it's a segment of the DNA, and that's the segment that we're focused on with transcription, the transcription unit, because it contains the information, in eukaryotes anyway, for a single gene. In bacteria, it may contain the information of multiple genes, all clustered together in this one piece of the DNA. Okay, and in bacteria, when we see this, that's called an operon. Okay, operons, and we're going to get to those a little bit later on. Okay, but in terms of RNA, what is going on with most of this RNA? Well, most of the RNA that we're making, it's actually rRNA, ribosomal RNA. And, even though mRNA has the most popularity because it directly results in proteins, mRNA only makes up about 3 to 5% of the total mRNA, or of the total RNA that's made. Okay? And, but there are still thousands of different mRNA species, worming around inside of the cytoplasm. And, on average, though, if you look at each individual species of mRNA, there may only be 10 to 15 molecules at any given time floating around somewhere in there. Okay? All right, let's look at the signals that are encoded in the DNA that tell the RNA polymerase where to stop, where to start and where to stop. Okay, so this sort of goes back to the idea of the transcription unit. Okay, well, first off, let's start with bacteria. Okay, now the bacterial RNA polymerase, just called RNA polymerase, is a large multi-subunit protein, right? It's an enzyme, but it has a detachable sigma factor. And, if we go through the basic steps of how it works, the first thing that happens is the sigma factor recognizes a region of the DNA in front of the gene, and that region is called the promoter. And there are certain sequences in that promoter that help the sigma factor to find it. And we'll get to that. But the second thing that happens is that the RNA polymerase sticks to the promoter. It binds to the promoter. And then thirdly, the DNA becomes unwound. And there's an enzyme that takes care of this, and it doesn't require any ATP hydrolysis. No energy goes into it, and we'll talk about how that happens. And fourthly, after the first 10 or so nucleotides, then the sigma factor releases from the RNA polymerase, and then the RNA polymerase sort of switches gears, right? It switches gears and undergoes a structural change and it lets it move much more rapidly. It just takes off. And we call this chain elongation. And how fast does it go? It cruises along at about 50 nucleotides per second. Okay, and then the RNA polymerase reaches the terminator. And when it gets to the terminator, then it's going to release the DNA and the transcript. And then the free RNA polymerase will reattach with the sigma factor, and it's on its way to find yet another promoter to start the process all over again. Okay, let's take a quick break before we move on.
We don't need no education. We don't need no thought control. Dark sarcasm in the classroom.